I want to call this forum together, and I really want to welcome everybody. As I said earlier, sort of sotto voce, it's great to see so many people here and interested in the subject matter. Today, our forum is entitled Preventing and Treating Breast Cancer, Seeking the Right Treatment at the Right Time. Sounds simple, but I've learned through my family it's very complicated. So we very much look forward to hearing our speakers, and I have been just delighted to be able to work with Senator Hutchison throughout her career on many different subjects, but this is really a good one for us in her final year in the Senate. I have become very interested in cancer because as I grew up, it grew, and it appeared to me to be all around me. My husband's father died when he was 10 of breast cancer. His mother died in her 70s of breast cancer. My stepdaughter has breast cancer. She doesn't apparently have the gene, but nonetheless, she has breast cancer. And so all around me, friends, we've gone through this. And so it remains an issue of great concern to me. And when I came to the Senate, I determined, well, if I can do anything to be of help, I want to do it. Breast cancer, as you all know, is the most frequently diagnosed cancer in women. It's the second leading cause of cancer death in women after lung cancer. We now know there is not one kind of breast cancer, as I thought in my young day, but 5 different subtypes with distinct biological features. What has changed is the recognition that these distinct biological features respond differently to different treatments. Therefore, the unique characteristics of each woman's breast and even a man's breast dictate which treatment course will be the most effective. Disease characteristics include the stage, the size, the grade of tumor, whether or not lymph nodes are involved, whether the tumor is hormone positive or negative, and whether the tumor is positive or negative for the protein HER2, the human epidermal growth factor receptor. In 1975, the only accepted surgical option for breast cancer was a mastectomy. My best friend had a double mastectomy around that time, and she's still alive and doing well. Today, the preferred surgical option is a lumpectomy, followed by radiation therapy. Then, depending on the type of breast cancer, a myriad of treatment options exist, including hormone therapy, drug therapy, and combination chemotherapy. In 1975, there were no genetic links to an increased risk of breast cancer. Today, several genes that make a woman more susceptible to breast cancer have been identified. As research continues, scientists will use additional information to identify changes in genes that may predict recurrence of breast cancer. They will also be able to predict metastasis, the spread of cancer to the rest of the body, and what treatments result in the best outcomes. The Cancer Genome Atlas at the National Institutes of Health is mapping out the genetic changes of 20 cancers, including breast cancer. Along with treatment, we've learned more about prevention and the effects of diet and exercise on cancer risk and reoccurrence. While there's no doubt that tremendous achievements have been made in cancer research and care, the costs continue to rise. Critical advances in treatment mean that now more than ever, though, women can survive breast cancer. However, while the many treatment options available, I believe there is a risk of confusion in newly diagnosed women and their families about which treatments are appropriate for a particular kind of breast cancer. And this has gone on in my family. Should she have a lumpectomy, a mastectomy, radiation, drug treatment? this drug is bad for her. She can't tolerate that drug. Chemotherapy, a combination of treatments. So it's my hope today that our forum today will shed some light on which treatments may be indicated for a particular kind of breast cancer, particularly in light of genetic tests and advances that have resulted in much more personalized treatment plans. So now, with great delight, it's my pleasure to turn to my distinguished co-chairman, the great senator from the state of Texas, Kay Bailey Hutchison. And then I'll introduce the witnesses, and Kay will introduce one.
Well, thank you so much, Senator Feinstein. It is wonderful to co-chair again with you. We have been chairman and ranking member, respectively, of the Military Construction Committee of Appropriations, and always had a great working relationship. And I want to commend also Senator Feinstein, not only for her just ongoing effort to do the most that we can in all cancers. Obviously, this specific one is personal to her, but the breast cancer awareness stamp that was Dianne's idea, and we were able to get that stamp approved and renewed, and we have, through that stamp, voluntarily people buying it and paying just a few pennies more, we have put millions into breast cancer research.
Actually, it's well over 60 million now.
60
it's
million.
Gone from the stamp to breast cancer.
I knew that you would know. I was hoping that was your cue. 60 million, and that was her idea, and I was proud to be her co-sponsor on that bill because all of this research that we are doing is giving us the treatments that Dianne mentioned, the variations in treatments. What can be best for one person is different for another because we now know there are different kinds of breast cancer. And the molecular makeup of the cancer itself is one of the great breakthroughs that the research that we have put federal money in. We have the volunteer sector, we have the cancer organizations like Susan G. Komen that also have put millions in. And all of this has led us to better treatment. And I will say the cancer organizations have done so much for early detection, just making people aware that they need to be looking for it. Catching it early has saved countless thousands of lives. So I am very pleased now that we're going to have this panel that will talk about the findings that we have and what it's doing for us. But I just want to say, as I'm in my final stages of my Senate career, that I have been a champion of all research, scientific as well as medical. It has been one of the focuses of my whole Senate career. And now we want to take the next step. Excuse me. We want to not only detect and treat well and prolong life, but let's eradicate it. Let's do what Salk did with polio and not have it anymore. Mostly anymore, have a little bit of an uptick now, but as long as we can get a vaccine that will prevent or know what the cause is so that we can prevent it, that would be even better, and only research will do that. So thank you so much for calling this hearing, and
thank you.
Let's hear from the witnesses.
Thank you very much. I'm going to ask the witnesses to try and confine their statements to 5 minutes. Unfortunately, we have a farm bill on the floor and 75 amendments to begin. So this is important, and I hope the witnesses, this is not a scientific meeting.
Oh.
Yeah, I know you're disappointed. So I hope that you will talk in simple language that people like me can understand. So now let me welcome our distinguished witnesses. Dr. Barbara Wold joined the National Cancer Institute to found the Center for Cancer Genomics in September of 2011. She's leading this new center for 1 year while she's on leave from her position as the Bren Professor of Molecular Biology at the California Institute of Technology, better known as Caltech. She recently completed a decade as the director of the Caltech Beckman Institute, which is dedicated to cutting-edge technology development in biology and chemistry. She began working on genome structure and gene regulation for her PhD thesis at Caltech, did postdoctoral work on gene function at Columbia College of Physicians and Surgeons in New York, and returned to the Caltech faculty in '81, where she charted the architecture of gene networks. This included developing modern DNA sequencing methods to map regions of the genome. Barbara, if you would begin, and I'll interrupt you at the 5-minute point, if that's all right. Oh, down the other end. All right, thank you.
Yes. So the mission of the Center for Cancer Genomics at NCI is to develop and apply cutting-edge genome science to prevent breast cancer and to better treat cancer patients. And I think it's important that the cancer patient is right in the mission for a cutting-edge research enterprise such as this. Cancer is many diseases, yet all of them have a common origin. The DNA of tumor cells is mutated or modified in the genes that govern cell division, survival, migration, and a number of other important cellular functions. These DNA changes do their dirt, they do their harm, by altering the amount, the structure, and the function of RNA and protein molecules, which are the active products of our genes. There have been spectacular improvements in DNA sequencing methods over the past decade, and these now allow us to determine exactly how an individual tumor's DNA has been mutated. The path of discovery, this path, has important implications for better prevention, for better diagnosis, and ultimately for better treatment of cancer. This morning, I'd like to tell you a few key lessons that we've learned by systematically applying modern genomics to breast cancer. And I also want to point out how parallel research on other types of cancer is yielding valuable insights into breast cancer and vice versa. Breast cancer, as the senator pointed out, is one of the most common cancers, with almost 230,000 cases and 40,000 deaths each year in the U.S. Research over the past 3 decades has taught us that breast cancer, like the other adult human cancers, is caused by multiple mutations in the DNA of a cell that ultimately becomes a tumor. So this is a long process over many decades. Most of the pertinent DNA mutations occur by chance, and they accumulate over a period of many years, although in some patients, as we know, one of the mutations is inherited from a parent, as is the case for breast and ovarian tumors with inherited BRCA1 or BRCA2 mutations. We're presently learning an enormous amount about which gene mutations can cause breast cancer as part of the Cancer Genome Atlas, also called TCGA. TCGA is the flagship program in adult cancer genomics, and it was developed to run in a close partnership between NCI's CCG and the National Human Genome Institute, called NHGRI. So TCGA is going to be complete in 2014. Cancer genomics won't be complete in 2014, but the TCGA program will. And it's creating a reference genome and genome-related data for more than 10,000 tumors. These 10,000 tumors include 20 major adult tumor types and 10 rarer ones. Each TCGA tumor is characterized by multiple genomic methods in a systematic and rigorous way, and the resulting reference data are made available to all cancer researchers. And this is important. This is a reference that people can go to, researchers around the country and around the world. Another program in CCG is called TARGET, which stands for Therapeutically Applicable Research to Generate Effective Treatment. And it leads a similar effort to TCGA in the childhood cancers, with a special emphasis on tumors that respond poorly to the current therapies. So one of the general lessons that's been learned from work on many cancers is that combinations of mutations in a small number of genes are needed to produce a tumor. We also know that all breast cancers are not identical at the DNA level, very far from it. So every tumor actually has its own genomic personality, and each one falls into a subset, as was mentioned earlier, of breast cancers with some commonalities and also some distinctions from each other, subgroup by subgroup. And within those subgroups, there are even distinctions that we're now picking out at the DNA level. So for example, we've known for a long time that excess HER2 protein caused by extra copies of the HER2 gene is associated with a particularly aggressive group of breast tumors. And knowing this about HER2 led to the development of Herceptin, about which Dr. McCormick will say more, which is a drug that provides an effective targeted treatment for many HER2-positive tumors. As is the case for other targetable so-called driver or causal genes, further research is now identifying new and additional drugs directed at HER2-positive tumors. And what that'll do is to expand the suite of options available to physicians. You can almost think of it as a spice rack with multiple spices applicable to a given problem. And the more that the doctor can pull out in the way of variety of treatment, the more hope one has of dealing with resistance to drug treatments and so forth. At the genomics level, the TCGA consortium confirmed 10 previously known breast cancer genes, which is what such a study should be able to do, confirm prior things. But it also identified 10 new and novel ones. So these studies are showing us that specific drivers found in one tumor type can also be active in tumors from unrelated tissues. And I think that's an important conclusion. So for this reason, it means that studying tumors of one type often enriches what we know about another tumor type, and in unpredictable ways. We don't know what we're going to discover until we learn about these tumors and learn about large numbers of them because they differ from each other and are so personal. We expect that these relationships are going to change and refine diagnostic categories and suggest different and effective uses of existing drugs, as well as propel the development of new ones. Learning the specific identity of a driver gene set for an individual tumor is ultimately powerful in the clinic because it can help the physician to assess whether a given drug will be effective in a particular patient. So we already do this for estrogen sensitivity and HER2 status in breast cancer and with EGFR status in lung cancer, for example. And the list of these distinctions that we can make is growing very fast, making it exciting times. Genomic knowledge can also be used to decide against a particular treatment if the appropriate target mutations are not in play. And I think this is incredibly important because in this way, you don't waste the patient's valuable energy and time, that precious therapeutic window, which can be short for cancer patients, on a treatment that doesn't fit the tumor. Indeed, some of the most exciting clinical research that's going on around the country at NCI centers and elsewhere uses extensive genome sequence information to guide an individual patient's treatment. Learning the DNA signature for each patient is ultimately going to be an important and routine part of diagnosis and treatment. So thank you very much for the opportunity to testify today. And I would like to thank all the patients who participated in TCGA and Target and other studies like it for donating their information and their biospecimens in order to propel the research forward. This is an information-intensive enterprise, and we need information from more and more patients going forward. And I'll be happy to answer any questions.
Thank you very much, Barbara. And now sitting on Barbara's left is Zora Brown. Zora is a 31-year survivor of bilateral breast cancer. She's an ovarian cancer survivor and is now fighting her third round with the disease. She is also a senior advisor of external affairs at Integris Health. She was founder and chairperson of the Cancer Awareness Program Services, CAPS, which educates women, particularly minority women, about cancer, and the Breast Cancer Resource Committee, which focuses on reducing the mortality rates of breast cancer on African American women. She's an outspoken advocate for minority and women's health issues and has received countless honors and awards for her advocacy and community service. She has previously served as a member of the Special Commission on Breast Cancer and the National Action Plan on Breast Cancer. And she's the lead author of a book in its 3rd edition, 100 Questions and Answers About Breast Cancer. Sora, we're delighted you could be with us, and please go ahead, and we look forward to your testimony.
Thank you, Senator Feinstein and Senator Hutchinson, for resurrecting this much-needed forum. And I also want to thank you, since I've moved to Oklahoma, for that farm bill as well.
Oh, yes.
And for me, cancer was a journey that began before I was born. Both my great-grandmother and grandmother were diagnosed with breast cancer at a time when little was known of this disease, and there was little reason for hope. Mammography had not yet been invented, genetic factors were unknown, radical mastectomy was virtually the only treatment option, and survival statistics were grim. No one knew what caused breast cancer, so it was something to discuss in whispers. So much has changed and improved since then, and even during my own lifetime. We know today, for example, why cancer has struck 5 generations of women in my family. A genetic mutation of the BRCA1 gene, which predisposes us to breast and ovarian cancer. We also know that as African Americans, the women of the Brown family and others like us are at risk for more aggressive cancer that strike earlier and have higher mortality rates. Understanding my family and racial histories taught me to be alert and proactive about my health. And although this knowledge did not render me immune from breast cancer, it did facilitate its early detection. First in 1981, when I was just 32 years old, and then again in 1997, which helped me survive and take back my life. As a member of a high-risk family, my mother, my sisters, and I, and now our nieces and daughters, have come to understand that we have been given not a genetic curse, but a benefit. The benefit of knowledge and the inspiration to use that knowledge to address the challenges of cancer and to imbue other survivors with hope. Now, in the midst of my 3rd round with this disease, stage 3 ovarian cancer, detected in 2005, I know all too well what a serious adversary I face. But I also know how to be an advocate for myself, to arm myself with information, and to surround myself with support in the best that science has to offer, so that cancer doesn't have to be the victor. And as a result, I continue to thrive day after day. Early on, I seized the opportunity to take part in a clinical trial that led to the identification of the BRCA1 gene, and most recently, I participated in a trial for patients with the BRCA gene, which is now not only allowing me to benefit from cutting-edge treatment, but to contribute to research that will advance the science around cancer and benefit future women like myself. At one point during my treatment, I had a 2 to 3 hour infusion every day for 5 days. The following week, I was infused for 2 days. I'm currently taking daily oral chemotherapy. The oral chemotherapy treatment has lifted an enormous burden of the anxiety of lost hours at work, among other things, and I feel much more productive. However, the cost and equity of oral versus infused chemotherapy creates sticker shock for many patients. Let me conclude by stating that the threat of cuts in research funding, not just for cancer, but all medical research, is alarming, and we can't allow the advances that may lead to a cure for this and other diseases to propel. Without the money required to pay for research and development, I might not be sitting before you today. The difference between death and survival may well be the ability to expand our knowledge of this disease through research. And I suggest that the threat of cuts in medical research funding is a huge problem, and we need your help in finding a solution. And while we may not have yet discovered a cure for our cancer, our crusade against this disease is far from lost. As a result of research, we have witnessed unprecedented advancements in cancer prevention and treatment. Because of research and clinical trials, we now know that cancer is a clinical illness that can strike anyone. Thousands of individuals who in the past would have concealed their illness are seeking risk reduction measures and treatment. Our army of researchers has isolated the BRCA1 and 2 genes, a discovery that holds enormous promise for the early identification of breast cancer in high-risk patients. We have expanded treatment options for breast cancer. Women are no longer exposed to the agony of undergoing biopsy and, depending on the findings, awakening to discover that one or both breasts have been removed. Patients can now choose lumpectomy with radiation as opposed to mastectomy. Bilateral prophylactic mastectomy is yet another option for consideration, and for particularly high-risk women. And depending on the case, many women have the option of foregoing surgical treatment altogether in favor of drug therapy. The research from which these developments spring go on with increased intensity, even as we gather here. In the United States, we enjoy the distinction as being people who, if we see a problem, we find a solution. Resilience defines us. For me, this has been a long journey, fortunately a long and slow journey, and I'd like to continue on it for a while.
right.
So thank you for all you do to help us get to that cure much faster. Thank you.
Oh, thank you, Zora. We're all on that journey with her, right? So thank you very much, yes. And now someone from my home stamping grounds, Dr. Frank McCormick. He is the president of the American Association for Cancer Research and the director of the University of California, San Francisco, Helen Diller Family Comprehensive Cancer Center. He holds the E. Dixon Hayes Distinguished Professorship in Oncology and the David A. Wood Distinguished Professorship of Tumor Biology and Cancer Research at University of California, San Francisco. He's the Associate Dean of the UCSF School of Medicine and Distinguished Professor of Microbiology and Immunology. That keeps you busy. Also biochemistry and physics, if that isn't enough. He pioneered cancer research by looking at the molecular basis for cancer. Previously, he founded the biotech company Onyx Pharmaceuticals and developed the cancer treatment Nexavar. He's received numerous awards and accolades in the field of cancer, chairs multiple task forces and committees, and is an elected member of the Institute of Medicine of the National Academies. Welcome, Frank. We look forward to your comments.
Well, thank you for your kind introduction. I'm Frank McCormick, Director of the UCSF Helen Diller Family Comprehensive Cancer Center and President of the American Association for Cancer Research. As President of the world's oldest and largest cancer research organization, I'm very honored to appear today to talk about the progress that we have made in saving lives from cancer and the unprecedented scientific opportunities that lie before us in cancer research. This really is a unique time in history when, for the first time, we really understand cancer at the molecular level. And although it's extremely complicated and presents many challenges, as Barbara alluded, it also presents many major opportunities for breakthroughs. Funds provided by Congress over the past many decades to the NIH and the NCI have made this extraordinary progress possible. And as influential leaders in Congress, you are to be commended for your consistent support of the NIH and the NCI. As a scientist whose own lab and whose cancer center depends heavily on the NCI for support and for its continued support, I'm personally extremely grateful. At the most basic level, cancer is a biological process gone awry. It is much more complex than we could possibly have imagined when the National Cancer Act was signed 40 years ago. In 1971, it was presumed that cancer was a single disease and perhaps there would be a single cure. This turns out to be not the case. Cancer is probably at least 200 different diseases. Cancer is caused by thousands of mutations in a very complicated manner, as we've already heard from Dr. Wold. Each of these different types of tumors requires different types of treatment. For example, we've learned that there are at least 7 different types of breast cancer, and with further research, we're likely to discover there are even more. Because we know now that breast cancers are not all the same, we understand why not all breast cancers can be treated the same. To capitalize on this knowledge, we must leave behind the era of one-size-fits-all cancer treatment and move swiftly into the realm of targeted treatments and personalized or precision medicine. This means that we treat patients based on the specific characteristics of the individual patient, on his or her tumor. This is one of the most promising new approaches in modern cancer treatment, and this is really what modern cancer treatment is all about, identifying exactly the molecular basis of an individual's tumor and targeting a specific therapy that will be most effective against that individual patient. To best illustrate for you the progress we have made across all cancers by telling you the story of trastuzumab, known by its trade name of Herceptin, a drug which has changed the lives of countless women with breast cancer since it was approved by the FDA in 1998. You've already heard something about Herceptin today, but I'd like to discuss further how Herceptin exemplifies the advances we have made in this field. This is really one of the first examples of targeted therapy and precision medicine. The roots of Herceptin's success can be traced to our fundamental understanding of oncogenes and cell signaling networks that led to the study and characterization of a receptor called HER2. This is research done in academic centers, supported mostly by the NCI all over the country. HER2 was shown to be present at high levels in human breast cancer tumors. We've since learned that about 20% of breast cancers involve overexpression of HER2, and these cases tend to be more aggressive and therefore more deadly. Further studies suggested that interfering with HER2 activity with an antibody could stop tumor growth, again, studies done in academic centers. More advances in the independently developed field of therapeutic antibodies, mostly in the private sector, led to the creation of Herceptin. And this therapy, which was novel at the time, showed dramatic positive results in clinical studies of women with HER2-positive metastatic breast cancer. Following its initial approval, Herceptin was approved in 2006 for use after surgery for early-stage HER2-positive breast cancer. In these cases, it was shown to reduce the risk of recurrence by up to 24%. The impact of Herceptin has continued to grow with the discovery that the therapy may benefit patients with the cancers of the esophagus, uterus, GI tract, bladder, and B-cell acute lymphoblastic leukemia, in which we see the HER2 gene overexpressed. For example, 2 years ago, Herceptin was approved by the FDA for use in patients with HER2-positive advanced gastric cancer. But the story doesn't stop with Herceptin. A continued pursuit of improved outcomes for women with HER2-positive breast cancer has led to a new therapy, pertuzumab, which was just recently approved by the FDA. And there's a whole range of derivatives and second-generation versions of these therapies which are currently being developed and are in the clinical pipeline. So as you can see, by continually probing the biology of cancer, researchers can make new discoveries and then apply that knowledge to improve clinical outcomes to patient benefit. I'll close by stating that if we are to fulfill the extraordinary results, extraordinary scientific and medical promise of cancer and biomedical research, it is going to require steadfast commitment from everyone, especially from the President and members of Congress, for sustained growth in the budgets of the NIH and the NCI. The 1.6 million Americans who received cancer diagnosis this year, including more than 230,000 women with breast cancer, are counting on us to continue the fight. Thank you. I'll be very happy to take any questions.
Thank you very much. Thank you. Dr. Leslie Bernstein is professor and director of the Division of Cancer Etiology within the Department of Population Sciences, big title, at the Beckman Research Institute and the City of Hope Comprehensive Cancer Center. She also serves as the Dean for Faculty Affairs at the City of Hope. An internationally recognized epidemiologist, she has focused her research primarily on breast cancer, seeking to identify modifiable lifestyle factors that will reduce and extend survival. She was the first to show a link between exercise activity and lower breast cancer risk. Dr. Bernstein has published more than 500 research papers and 50 book chapters during her career. She leads the California Teachers Study. That's a prospective cohort of over 134,000 female public school professionals formed to study breast and other cancers and actively involved in research collaborations examining the genetic and environmental risks for breast cancer. She's a recipient of many awards, including the KO5 Award from the National Cancer Institute. I want to welcome you here today, and we look forward to your comments.
Thank you so much, Senator Feinstein and Senator Hutchinson. It's really a pleasure to be here. I want to thank everyone who has brought this meeting together. City of Hope appreciates your continued leadership to support federal funding for cancer research and treatment. City of Hope was founded in 1913 and is a pioneer in the fields of bone marrow transplantation and genetics. I'm hoping it'll be a pioneer in cancer prevention now that I am there and working there. I have been there for now 5 years. It's a leading research institution, treatment institution, educational center for cancer, diabetes, and other life-threatening diseases. And we're located, for those who do not know where we are, in northeast of Los Angeles in Duarte, California. And we are a comprehensive cancer center. I started my career when I was in my 40s. I got a PhD at that time and began to work in breast cancer. And my goal was really to find novel factors that would reduce women's risk of breast cancer. And to do this by understanding some of the biological mechanisms that influence risk, and then most importantly, by finding practical ways, things we could tell women to do to reduce their risk of breast cancer. During that time, I met wonderful women like Zora, working with the National Breast Cancer Coalition, and so it also altered what I was doing, not just to find causes of breast cancer, but how to extend women's lives who had breast cancer. To begin to work in survivorship before survivorship was really on the table as an important thing to do. I began my research by trying to understand the hormonal basis for breast cancer. This was in the early 1980s. And you base things on your own experience. So I'd been a competitive swimmer who trained 8 hours a day as a high school student and experienced menstrual cycle disturbances. And we already knew at that time that when a woman had her first menstrual cycle or when she had her last one at menopause were important breast cancer risk factors. We also knew a little bit about women who were likely to develop breast cancer, like when they had their first babies. This was important. But we didn't have practical solutions because we couldn't recommend when you should stop menstruating or when you should have your first child. And so I began to study physical activity. It was my own experience. We knew that physical activity impacted menstrual cycles of young teenage girls. We knew that it actually regulated hormones that women were exposed to that come from the ovary. And so I began to study it and spend most of my career in the 80s and 90s trying to understand this, and was lucky enough to be one of the first people to link exercise to breast cancer risk and to show that, in fact, women who were physically active had much lower breast cancer risk. We did studies of young women who were in their 40s or younger when they were diagnosed with breast cancer. We studied postmenopausal women. We studied Asian American women. We studied African American women. And each of our studies showed a marked impact of physical activity on breast cancer risk. So that's one of these modifiable risk factors. There are others, and in the mid-1990s, a group of colleagues, epidemiologists, we all work in collaboration because trying to do prevention research requires a team. We formed the California Teacher Study, so it's a resource for California, and I am lucky enough to be the principal investigator for this large study. And we've continued to look for novel risk factors in the California Teacher Study. Factors like whether passive smoke exposure impacts breast cancer risk. And we've been able to confirm and extend our understanding of other factors like alcohol intake and breastfeeding, learned about how menopausal hormones influences some of the subtypes we've been talking about, the different subtypes of breast cancer, and we were able to show using the California Teacher Study how that impact changed, the rates changed depending on what type of cancer you had, that hormones affected only certain types of breast cancer. There's some interesting areas I hope we'll be pursuing, many of us in the future. Aspirin is one of them because it reduces inflammation, but it also inhibits aromatase. And aromatase inhibitors are drugs that are used to treat women with breast cancer. So women don't want to exercise if they haven't been doing it. We need to find more practical ways. They're not marching along taking tamoxifen as a prevention or taking raloxifene as a prevention drug. So I'm hoping, and before I finish my career, that we might learn that aspirin as an anti-inflammatory and an aromatase inhibitor might work. So in the meantime, we'll be working to try to understand what influences breast cancer and what practical messages we can give to women. One of the things we've found is, in fact, that these practical messages also work for breast cancer patients who are post-treatment. I appreciate the Senate Cancer Coalition's time to showcase the progress that we've made, and I'm very thankful that you're even considering prevention, as most meetings only consider treatment, and so thank you for letting me highlight this.
Thank you. And now I'd like to ask my co-chair, to recognize the final distinguished witness.
Thank you. Dr. Ana Maria Gonzalez Angelou attended medical school in Colombia and moved to the United States to continue her training in New Orleans and then to the University of Texas MD Anderson Cancer Center to specialize in breast cancer. She was a Susan G. Komen Breast Cancer Fellow. She is on the faculty there in the departments of breast medical oncology and systems biology, working exclusively with breast cancer patients and in breast cancer research. She is the chair of the Endocrine Resistance Working Group and a member of the Correlative Sciences Working Group for the Translational Breast Cancer Research Consortium. She serves as a member of the steering committee of the U.S. Middle East Partnership for Breast Cancer Awareness and Research and the Partnership for Breast Cancer Awareness and Research of the Americas. So she is the Texan on the panel. Welcome.
Senator Feinstein and Senator Hutchinson, thank you for giving me the opportunity to appear for you today. And thank you for the kind introduction. So I'm here to summarize in brief how we treat breast cancer in 5 minutes. So that's my challenge.
Oh, good.
Just briefly, the management of breast cancer has significantly evolved over the time, and with the incorporation of new treatments and better understanding of the disease itself, things have significantly improved. As Zola said, we started with a single option, which was radical mastectomy. And now with the introduction of radiation therapy, chemotherapy, targeted therapies, things have significantly evolved. So today, we make treatment decisions for patients with breast cancer based on their disease state, the disease subtype, like we described, and the risk that those patients have. So we divide the stages into 1, 2, 3, and 4. Stages 1 through 3 are what we call early breast cancer, and we can cure that disease. Unfortunately, stage 4 today is no curable disease. However, due to the advances that we have, patients live for long periods of time with very good quality of life. And in practical terms, although we have genomic technologies that have explained to us how biologically we have different subtypes, 5 or 6 subtypes of breast cancer, in practical purpose in clinic, we divide breast cancer in 3 subtypes. One subtype that expresses the hormone receptor, so the estrogen and progesterone receptor, one subtype that expresses the HER2 or HER2 receptor, and one subtype that doesn't express any of the 3 receptors that we call triple negative, and is probably today one of the most aggressive type of breast cancer. So basically, when we talk about risk after treatment completion, we talk about the risk that the cancer has of coming back. And based on that risk, we choose the treatment for our patients with breast cancer. So we estimate the treatment based on the different characteristics of the tumor, how big the tumor is, the number of lymph nodes that are removed at the time of surgery, and they have cancer when they remove the lymph nodes from the axilla. We also look at these receptors that we talk about, the grade of the tumor, which is how ugly a cancer looks under the microscope. We assess that, and we estimate if a patient needs or doesn't need chemotherapy, for example. So in general, breast cancer treatment, we can split it in 2 parts. One part is called the local control, and that is what prevents the cancer coming back into the area of the breast or the areas that are close to the breast. And that's made of 2 parts, radiation therapy and surgery. And the other part is what we call systemic control. And systemic control is what prevents cancer coming back in other areas of the body. When cancer metastasizes, it tends to go to the lungs, the liver, the brain, the bones. And that is made out of endocrine therapy or anti-hormone therapy, chemotherapy, and targeted therapies such as trastuzumab or Herceptin. So when we talk about local control, we have different options for patients. We have either mastectomy or lumpectomy, and today, less is more. So our treatment of choice today, as senators say, is lumpectomy. And before, we used to remove all the lymph nodes in the armpit. And now we do a procedure that is called sentinel lymph node biopsy, which is to try to identify the first lymph node in which cancer has gone. And if that lymph node is clean, we stop right there and we avoid removal of the lymph nodes and the risk for the patients to develop lymphedema or those swollen arms that they're afraid of. There are some cases in which we have to do mastectomies. For example, when patients have more than one cancer in the breast in different quadrants. So that is an indication to do mastectomy. The other part of the local control is made out of radiation therapy, and there are specific indications for radiation therapy. For example, every patient that gets a lumpectomy requires radiation therapy. Every patient that has large tumors requires radiation therapy, and every patient that has a large number of lymph nodes, 3 or more, that are compromised require radiation therapy. The systemic control, which is the last part, is divided according to those 3 subtypes of breast cancer. So patients that have hormone receptor positive breast cancer should be treated with anti-hormone therapies. If they're premenopausal, we use a drug that is called tamoxifen. If they're postmenopausal, we use the aromatase inhibitors. Until about 2 or 3 years ago, every patient that had a tumor that was greater than 1 centimeter, even if their lymph nodes were negative, were treated with chemotherapy. But thanks to a genomic testing that we have today, we can actually define who of those patients actually benefit or don't benefit from chemotherapy, and we can avoid chemotherapy in more than half of those patients. We're doing the same type of clinical trial today in patients with positive lymph nodes, and we're trying to learn who are the patients that are at enough risk that require chemotherapy, and who are the patients that don't have benefit or they will not benefit from chemotherapy. Patients with HER2-positive breast cancer have to be treated with trastuzumab. Trastuzumab reduced the risk of recurrence and death for those patients significantly, and it went from being the most aggressive type of breast cancer to actually one of the breast cancers that today we can cure. Our big problem today is triple-negative breast cancer because we don't understand clearly the biology of that disease. And when we study this disease in the lab, we've seen that there are about not only one disease, but there are 6 different subtypes of triple-negative breast cancer. So this is, today, the only current treatment for triple-negative disease is chemotherapy. It is sensitive to chemotherapy. We can cure about 1/3 of those patients with chemo. However, we have 2/3 of those patients that succumb to the disease, and this is where our research should be focusing today. Thank you for the opportunity.
Thank you very, very much.
me to
I think we've had a very distinguished panel. I just mentioned to Senator Hutchison, I'd like to do something a little different today. I'm going to ask one question. She will ask one question. Then I'm going to ask our audience if you have a question that you might like to address to any one of our witnesses. It must be on the topic, however, or my bicep will come after you. Okay? So let me ask my question, and let me ask this of Zora Brown. Zora, we had one of these hearings, it was many years ago, and I wanted a cancer survivor, and so a young African-American woman testified. She was from New York, and what she spoke to was getting the diagnosis, not knowing where to go, what to do, being alone, having no one to consult with. She was imprinted on my mind. I think this was 10 years ago. And therefore, we began to think in terms of a patient quarterback, an oncologist. Should healthcare provide for coverage of an oncologist that goes with the woman every step of the way and gives her advice as to what to do? How is your experience different from what it was 30 years ago? When you first got the diagnosis, what options did you have? You know, what loneliness did you feel? What happened after that? If you could sort of trace that aspect for us, I think it might be very useful.
Thank you. As I started out by saying that this was a family journey, so my activity was a little bit different because I did have a family, and that's why I started the Cancer Awareness Program services, to be able to intervene for other women. What we have now, the improvement what we have now, is that we have patient navigators almost within every single facility that treats breast cancer and other cancer patients. That has helped to navigate the waters, if you will. And one of my friends calls it navigating the minefields, because there's so many questions. And I think when a woman is diagnosed with breast cancer, her mind just goes blank. You know, so she doesn't have the faculty to be able to maneuver all the things that she needs to know. led me to write 100 questions and answers about breast cancer. Also, giving women the tools that they need early on. I mean, when you're first diagnosed, everyone, I think, should be prescribed a support group. I really do. I think that whether you engage in it initially, eventually every woman seems to get into a support group. Sharing those commonalities has been one of the benefits that I see since before I was diagnosed. I had my family to rely on, and of course they gave me all the support I needed. But at the same time, when you leave home and you're out by yourself, you sometimes want to be able to grab on to someone else who's successfully gone through this process with you. And that's where I see us now. We've now come to the point where we have people who can talk to you, take you by the hand and sort of take you through that maze of what you need to know about breast cancer. We've come a real long way. So.
Thank you very much. Senator Hutchison?
Yes. My mother had uterine cancer, and she went to MD Anderson, had a wonderful doctor, and did very well. And this was when I was a teenager. And some 30 years later, when she was having a normal checkup, the doctor looked in her ear, just a general practitioner looked in her ear and said, you know, I see something growing there, and you need to go to an oncologist. Well, she had brain cancer that was the same as the uterine cancer, and this was 30 years later. And so what you said, Dr. McCormick, and what several of you have alluded to is that we have always treated the cancer where it is for our purposes of funding. So breast cancer, ovarian cancer, prostate cancer, and that's how we fund. But now this is all coming from my own experience, but also things that you have said. Really, it's a type of cancer which can occur in many different places. So my question is, should we be rejiggering the way we fund cancer now as United States senators on the strain of cancer? Can it be defined by types, and is that a better approach than the placement? Dr. McCormick, or really any of the...
Take a start.
Yes. Mm-hmm.
Well, that is a debate that's going on, I think, all over the country, because we see the same genes, the same driver genes can be responsible for very many different types of cancer. So you might think that therapies will be more driven by the actual driver mutation than the type of tissue that the cancer was derived from, in which will be a shift in the direction you're saying. And that is true for some examples we've already mentioned today. But there are other examples where that hasn't been the case. There have been, as you may know, dramatic improvements in treatment of melanoma over the last few years by recognition of a driver gene called BRAF, which causes 60% of melanoma. And a drug has been developed that is very effective against BRAF mutant melanoma. But very surprisingly, the same drug was not effective in BRAF-driven colon cancer, although most people expected it to be.
Yeah.
So we have to recognize that these driver genes function differently in different contexts. So I think we're not ready to abandon
Very
that.
interesting.
We're in that point where we're not, there's a case to be made for looking at how drugs that target a specific gene target different types of cancer, but I think it's premature to abandon the definitions by disease at this point.
I think Dr. Wald was looking like she had an answer as well, or
No, I would
Were you
just
just
like to
looking
endorse Dr. McCormick's answer and say perhaps one coda, which is as we see that every given tumor involves a couple of genes, the idea that the combinations are part of the context that he described that makes one tumor that looks on the basis of one of its mutant genes like it should respond like the others, not. And so this is going to be an evolution, I think, in our way of thinking, and perhaps the funding will follow. But I think...
thank you.
All right. Now the great experiment. In 20 years, I have never done that. Don't disappoint me out there. Does anyone have a question? If you'd begin by just introducing yourself and ask your question and indicate whom you might like to answer it. Yes. Could you stand up so we can hear you? Can anyone answer that? I don't. No, Dr. Wold, I think, can answer it.
Well, I think our hope of regaining it depends perhaps more with you than it does with me. And so I will toss it to the senators.
Well, as many know, we had a great mission earlier on to double the research and came very close to doing it. There is now an acute shortage of dollars. As a matter of fact, the conversation here is what happens next year when we go over a cliff where a lot of revenue stops. The payroll tax cuts go out, the Bush tax cuts end. There are many problems coming up. So this has to be a relatively conservative time. I think I can safely say that virtually every member of the Senate gives a high priority to medical research and really wants to see dollars for medical research. And I think that's the closest I can come. Senator?
I do agree. I think that a vast majority of the Senate values research and considers it a priority and an investment. We reauthorized the America Competes Act just last year, and that was unanimous, actually, in the Senate, and that is a research base. So I do think that that commitment is there. I can't tell you it's going to be $6 billion, but I can tell you that within the framework of fiscal responsibility, which we have no choice but to do, nevertheless, the areas that will be prioritized will include research.
Well, I'm going to ask a question because I don't see a lot of hands out there. Dr. Gonzalez, I thought you made a very good statement, and you detailed the variety of treatments available for a woman. You described both local control, systematic control. Can you just elaborate clearly and succinctly, because I'm still puzzled by it? When would you recommend a mastectomy versus a lumpectomy?
So today we try to avoid mastectomies in most of our patients. So our initial recommendation for a patient is to have breast conservation, which it's made actually of lumpectomy.
Even on a reoccurrence?
Hold on. So we started with lumpectomy and then radiation therapy. Now, once we have that, there are certain contraindications to the lumpectomy. One is, for example, to have more than one tumor in the same breast. So if I have a tumor, for example, in the upper outer part of the breast, and I have a second tumor in the lower inner part of the breast, the entire breast needs to be removed because it is a higher risk that there is a teeny tiny tumor somewhere else that we're not seeing and we can leave cancer behind. The other contraindication is, for example, a subtype of breast cancer that is a very aggressive and rare type of cancer that is called inflammatory breast cancer. And then the other group of patients in which we advise mastectomies are patients that have BRCA mutations, so have that gene that put them at risk because once they develop one breast cancer, they continue to be at risk to develop a second and a third and a fourth if they have breast tissue behind. Those patients, though, have the option of, if they want to preserve their breast, have very, very strict follow-up and screening to make sure that if they develop a cancer, we can cut it on time and they can be treated appropriately. The recurrence is a little different. When we have a patient that had treatment for breast cancer and had breast conservation and breast cancer came back on that breast, we cannot do breast conservation anymore because that breast had already radiation and we cannot re-irradiate the breast. So at that point, we do a mastectomy, we wait for healing, and we offer the patient to have a reconstruction.
Anyone like to add to that? Senator, you have another question?
Well, yes, I want to say that I thought that well, first of all, let me say, I thought every single person on the panel offered something different, and I really liked that. I thought Dr. Bernstein's sort of practical, what you can do in everyday life focus was excellent, and that you are looking at something so available as aspirin as a potential to look at that might be a deterrent. Are there other areas like that that are so readily available where if it has an impact, like if aspirin did turn out to be successful, oh my gosh, what an easy, simple thing. Are there other things in your arsenal that you could share with us that are practical and we could do every day for prevention?
Well, I wish it were an arsenal and that we had a multiplicity of things. One of the ones I failed to mention was obesity, which is really important both in developing breast cancer, but also in preventing recurrences later on, because we know obesity is one of the most important factors predicting shorter survival for women with breast cancer. Arsenal, You know, I think people look for simple solutions. That's why I'm trying to look at something as simple as aspirin. Metformin, a drug that is used to treat type 2 diabetes, seems to have some great promise, and other researchers are looking into that. But, you know, these are like magic bullets. The problem is, how do you convince a population to go out and exercise, maintain their weight, not gain, limit their alcohol intake, breastfeed their children? These are the things we know right now. But we're probably missing some things. When I came in, as I said, we only knew about reproductive factors, and then we began to learn about hormone therapy. We still, although oral contraceptives don't appear to have a marked impact on breast cancer risk now that the formulations have changed, we still study them because they keep changing the formulations. So I don't have more magic bullets yet, but there's a lot of work that's ongoing. People are interested in vitamin D, whether that might have an effect. So we're looking for those recommendations we can make, whether people will pick up on them, whether women will practice them. That I'm not sure. In terms of that question about, you know, should we study breast cancer, or are the factors that we look at more pervasive? We do that as well in epidemiology and find many of the factors that we study for one type of cancer actually apply to another type of cancer. So most of our studies look at more than one cancer.
Everybody's come awake. All right. Please stand and give us your name.
Thank you. I'm Marge Fody from the American Association for Cancer Research. I would like to ask the panel to address the issue of personalized therapies. We're obviously in the new era of personalized cancer medicine and precision medicine. What are the greatest challenges that we face in order to be able to bring new drugs, new treatments to patients? Is it funding? Is it regulations? Is it science? What are the issues that we need to grapple with at this point?
All right, that's a good question, and I'm going to ask our experts. I'm also going to ask them to add one thing to the treatment. Is a treatment different for a 35-year-old woman than it would be for a 55 or 65-year-old woman? Who would like to take that? Go ahead, doctor.
Since I'm the one that treated the
Yeah, don't be
45
shy
to
down at
55.
this end.
So yes, I think that breast cancer is one of the few tumors that today we can say that we do as much personalized therapy as we can, but we should be able to, we need to do better. So today we have the privilege of being able to, for example, have a genomic test that allowed us to see if patients will benefit from chemotherapy or not and avoid the side effects of the chemotherapy in a very large group of patients versus actually select the ones that need the specific chemotherapy. We also can give trastuzumab to the patients that have HER2-positive disease and cure that disease. But we have to put into context a lot of the information. For example, it's not the same to treat a very young patient, let's say a 25, 28-year-old patient that has triple-negative breast cancer, than a 75-year-old patient that has hormone receptor-positive disease. One will receive a lot of 6 months of chemotherapy. The other one will receive probably breast conservation surgery and a pill every day with no chemotherapy. And today we're actually questioning the need for radiation. So I think that we're getting better and better. I think that the limitations that Dr. Foti allows are we need to do better in science. We have cancer is like, I like to quote George Sledge, which was the former president of ASCO, that says that one dumb tumor is smarter than 10 smart oncologists. Yeah. And it's true. I mean, there's a lot of heterogeneity of the disease, and it's very hard to understand what the disease are, and the disease actually finds a way around of the treatments. So we need to do better sciences, and we need to study better and do that. And in order for that, we need the funding. The regulation part that Dr. Foti allows is also true. It's very difficult to being a translational researcher, it's very difficult to bring the discoveries from the lab to the clinic. And Dr. McCormick can tell that because he created a drug that is used today to treat several types of cancer. There's a very useful drug, but to bring those discoveries from the lab to the clinic take years and years of work and years of paperwork.
Yes.
Well, thank you. That's helpful. Dr. McCormick, respond.
Okay, well, well, developing a drug certainly is a very long and expensive and risky process, but the main problem really is the complexity of the disease itself, and identifying what the drivers are that really the tumor depends on for survival. We mentioned HER2 new in breast cancer, turns out to be a great target. BRAF in melanoma, a great target. So we know that's a driver of the cancer. If we hit that driver with a drug, we expect to see a clinical response. But in many cancers, we don't know what the driver is. And ovarian cancer, there doesn't seem to be one single driver. Triple-negative breast cancer, we don't know if there's a single major driver.
What's triple-negative breast cancer? Explain.
It's a definition that we've heard from Dr. Gonzalez. It's basically
So
estrogen-negative,
yeah,
testosterone-negative, HER2-negative. It's a clinical definition. Sorry.
Thank you.
It's one of the subsets of breast cancer which is the most difficult to treat, and partly because we don't know what the driver is. We have no way of matching a drug to the driver if we don't know what the driver is. In pancreas cancer, we know what the driver is, but we can't figure out a way of finding a drug to hit it. So there are many enormous gaps in our ability to actually deal with the knowledge that we already have. And that means more research to understand and to develop new innovative methods for attacking what's called undruggable drivers and for discovering drivers in tumors which we can't really currently understand. Would you agree with that?
Once again, I endorse everything that you said. And my coda this time is that our tradition of testing and approving drugs in a particular setting for a particular kind of tumor is a tried and true one, and one we should not abandon. However, off-label use, so-called off-label use, is now driven by the science. For when you see a driver in a type of tumor where a drug has not been tested, but it appears as if it's likely playing an important role, the doctor will probably be motivated, all other things being equal, to attempt to use that drug. But now you're in the realm of off-label use, and that has real implications in the structure of our healthcare system.
Thank you. Yes. Go ahead, doctor.
We're coming to your neighborhood with a big public event in July.
Good answer.
And it's aimed specifically from the point of view of TCGA for bringing up the participation and numbers of patient samples who are from the underrepresented minority community, particularly in this case in breast and prostate, but moving forward. And so there's a big and specific push in that direction in order to enable hospitals that more frequently treat larger numbers of minority patients to actively participate. Yep. Well, I mean, metaphorically, sort of your
Close
neighborhood.
enough. Anybody else? Yes, if you give your name.
I'm Dianne from the National Breast Cancer Coalition, and I wanted to address something that Senator Hutchinson brought up in her opening remarks about a vaccine. I know it's something that we're very interested in, and I know that there's a number of other groups participating. Dr. Bernstein, since you've been touching a lot on the prevention angle, if you could start, I'd be interested to hear from the whole panel about the idea of a vaccine for us.
Well, yes, I've been part of the Artemis project from the National Breast Cancer Coalition, which is looking at the possibility of developing a vaccine, not the kind of vaccine you think about when you think about the HPV vaccine or a smallpox vaccine, but a genetically driven vaccine that would prevent breast cancer. And so we are working with immunologists and other biologically oriented scientists, people who come from fields that we're not used to try to work this all out and see if it's possible. My role is simply study design because that's my expertise. So I sit there and listen to what they're doing, but the goal is before 2020 to have something like this in place.
Oh, that's great. That's great. Anybody else?
May I go back to the question about increasing minority participation in clinical trials? I think we need to increase participation in clinical trials across the board, but I work with the National Medical Association on Project Impact, and that is one public service activity where we've tried to get minorities to understand the benefit of participating in clinical trials. When I moved to Oklahoma and wanted to participate in a clinical trial, every scientist or community told me, well, you need to go to Loma Linda. Or perhaps you can get it to Dana-Farber, or maybe MD Anderson. And I'm like, look, people, I live in Oklahoma. I can't afford to get on a plane and go off for 2 weeks to get this therapy. So what we were able to do was to collaborate with the pharmaceutical company so that I could receive the drug. I got infused 5 days a week, and I went to work every day. So I know that there are people who want to participate, but just those barriers of not being able to get to your job, that's a big barrier for most minority women. And also where the trials are located. We have to figure out a way to collaborate more with community cancer centers so that we can get more people into clinical trials in a setting where they are comfortable.
Absolutely. Anybody? Yes. Oh, pardon me, what?
I just think one more comment about the vaccine issue.
Sure, go ahead, and we'll get to you.
So back to the vaccine kind of issue, I think it's extremely exciting to broaden that and think about genomically driven, happens to be, immune therapies. And so there are many of these strategies that revolve around the interface between the immune system, and particularly there may come a day soon, and I know there are some trials being organized that are literally around the subject of a custom vaccine for one person based on their tumor in order to attempt to help their immune system defeat it. And it's early days, but I think it's one of the most exciting genome interfaces in the research forward.
Yeah, go ahead, Doctor.
Yeah, I'd like to echo that.
Frank.
But also to point out that some of the most exciting breakthroughs in treatment of cancer have come from turbocharging the immune system just in the last few years. This has been an area which really hasn't borne fruit for many, many decades, but suddenly it's really sort of hit prime time. So the approval of Provenge for prostate cancer, but more dramatically, 2 new therapies that basically make the cancer cell more recognizable to the immune system have led to amazing durable responses in melanoma, for example, where patients seem to be alive and healthy after 5 years after treatment, which in melanoma is an absolutely astonishing breakthrough. And in lung cancer, also another immune-based therapy is showing really durable and amazing responses. So this field has really come to life, again, through research and innovation it's really been translated into real benefit.
Is there a possibility that if you have the BRCA, that there could be a vaccine that you could just take which would prevent the development of breast cancer?
That's based on
Or
what we
is
know.
that
Well, based on what we know
just
today,
what
I'd say that
thinking?
we couldn't think of a way of doing that today. But I think it's feasible that if we understood enough about the biology of those tumors, with some more innovations, we might be able to devise a therapy specifically for those types of tumors. We can't do it based on what we know today. Is that fair, do you think, Barbara?
I think that's fair.
Yeah.
Okay, anybody? Yes, I said, right.
I'm Lori Williams, and I'm a nurse scientist at MD Anderson, also on the board of directors of the Oncology Nursing Society. But I also know the risk of breast cancer.
Belt it out so everybody.
My mother died of breast cancer. And I wanted to ask Dr. Bernstein, because you mentioned that there are very few women who are using prevention that we know right now works, like tamoxifen and raloxifene. Do we know, is it because they never get it recommended that they take it? I mean, I went for my first breast cancer screening visit at MD Anderson. They looked at my risk factor, and I took raloxifene for 5 years. Why are they not taking it? And what makes us think that maybe they would take aspirin if they're not doing the other?
I think the issue about whether tamoxifen and raloxifene are being recommended, I would say not universally by internists and general practitioners who would be the ones to actually take the history and make the recommendation. But where they have universally recommended it, the uptake has been low. One of the complications for tamoxifen is if you have a uterus, it increases your risk of endometrial cancer. We did research on this and found that it was really specific to women who were heavier or women who had been on estrogen therapy before their breast cancer. But in fact, women fear these side effects. There's also the cost. And so not everybody has a prescription plan, and these drugs, they don't cost what aspirin costs. And so I think if women are looking for some means of preventing breast cancer that's easy and they want a pill, we know so much about aspirin. We have, decades and decades of research on it. We know what it will do. Most people have taken it. Most people can take it. I can't guarantee it does anything. This is the research I want to do. So I'm not recommending it. I'm just saying by its biological properties, it should work, but it might not.
Interesting.
Well, nothing is ever what we predict.
Thank you, yes. Doctor, why don't you begin and then we'll...
I will pass on that since I'm not an MD.
Depends on the subtype of breast cancer. If a postmenopausal patient or a premenopausal patient has, for example, triple-negative breast cancer, the standard treatment is chemotherapy. And we give the same chemotherapy to the older patient compared to the younger patient, unless there's a contraindication for specific medications. For example, if the elderly person has problems with their heart. So I have to avoid some of those drugs. For HER2-positive disease, it's the same. For hormone receptor-positive disease, it depends. Patients that are younger tend to have more rapidly growing tumors that are more sensitive to chemotherapy or require chemotherapy. Patients that are postmenopausal tend to have more slow-growing tumors, and sometimes endocrine therapy or hormone therapy is enough. So we just have to kind of personalize or individualize the type of patients that we have and make decisions about for the treatment, considering the risk, their age, and some other things.
Yes.
Hi, my name is Kate Bell, and I'm an oncology nurse and a University of Texas graduate fellow. And my question is regarding, so we talked a lot about prevention, and I was wondering from a public health perspective, particularly with minorities and Medicare underserved populations, where you see the efforts going to reach those populations in prevention.
Zora, you want to...
Actually, I don't see them. I really don't. I think that we have a lot of patient advocates who are doing their best to try to get more minorities into clinical trials, to educate them and what have you, but the funding isn't there. And so you have organizations that were run like mine for 20 years, and I was creating a path to the poorhouse pretty quickly, because you really do, because the funding just isn't there. And I think that's one of the ways that we're going to get more minorities and more people into clinical trials, is to recognize that the patient-to-patient advocacy and encouragement is going to make more people aware of what is needed, what's out there, and why it's important to be in clinical trials.
Anybody, would you like to?
Yeah, I think many of the cancer centers, the comprehensive cancer centers, are making these efforts into the communities and into the minority communities to try to educate more. It's a very difficult task, and we actually need members of those communities to work with us. We have volunteers. It's easy when you're working with breast cancer survivors and advocates, because they're wonderful and they'll do anything to educate other people. But we have to broaden these efforts, and they are difficult. But this is part of what we're supposed to be doing in terms of translating our research. It's getting it out to the communities as well as translating it to the bedside.
I'd like to just jump in, because we all have heard about the delays of the FDA in approving drugs. Does the panel think that the clinical trials are an offset to the delays that does make a difference? And if we needed to do any kind of reform of FDA, would you suggest that there be more clinical trials, or that we instead move the timelines, or urge the movement of timelines to be shorter?
Frank, you want to go ahead?
Yeah, well, one thing, timelines will be long when patient survival is an endpoint. And as more and more drugs approved, survival times of patients get pushed out. So every drug basically takes longer to assess in terms of its impact on survival. So time is an issue which you can't really get around very easily. But
The approval?
yeah, the
of
approval,
the
right, but
drug?
approval is generally based on time to progression or survival, and that's a matter which is measured can be measured in months or years. So that process will take time. But I think one of the big issues is that, as Barbara already said, we're now having to deal with combinations of drugs to be effective. And this raises all kinds of complexity about how the FDA will approve those kinds of drugs. So in fact, in my role as the president of the ACR, we have a dialogue with the FDA. We are going to the FDA in a few weeks, actually, to help work with them, to help work through the problems and the challenges of trying to address the complexities of clinical trials. They are very open and very receptive to new ways of doing things. They want to they want to approve drugs that have a big impact. And they're facing the same complexity and challenges as we are in the scientific community. So my sense is that the people at the FDA we talk to are extremely well-informed. They want to be more informed. They're reaching out to us to help educate them to the complexities of the issue. But, they would claim to be underfunded and overworked. I'm sure that's true. But they understand the complexities of the issues, and we in the scientific community are working with them to try and help navigate through. But it's a really tough issue. For example, you might say that measuring responses to drugs in short term will predict whether patients will survive long term. That's often not the case. Sometimes patients respond well to a drug but end up passing away on the same day, receiving no benefit in survival. So it's not a simple issue, and I think the FDA are struggling with that, and again, we as a community are working with them to try and navigate through those terribly complex issues.
Would you join with me? I have another appointment, and these people came a long distance to be here today. I think they were all quite wonderful. Would you give them a big round of applause, please? And Kate, can you stay? Okay. So let me add my word of thanks. And let me say, this is such an emotional, difficult subject for so many of us. The fact that you were really clear, I've done these panels before, and for me, this was the best. I learned the most, and what became very apparent, which is really should be heartening for all of us, where breast cancer stalks us one way or another, is so much is being done to find the answers and to help. So I hope everybody that leaves this room leaves it with that feeling, and that's all because of people, not Kay and myself, but the people that surround us. They're very special, and I just want to say thank you to each one of them for their role.
I
Thank you.
agree.
Thank you
and,
so much.
Again, I thought each of you gave a different perspective and added something so valuable. And I think the fact that the room is completely full of people interested in what you all were bringing to the table and interested in our working together to find the funding sources and make sure that we're doing the right thing in the right places. It's very helpful to us. So thank you all very
And
much.
thank you, and buy breast cancer stamps. Thank you very much.
Thank you so much. Yeah, great discussion, yeah.
Well, you're welcome.